Today's topic is T-cell receptors. T-cell receptors or TCR are membrane-bound glycoproteins present on the surface of T cells or T lymphocytes. They are the major molecules responsible for T cell specificity. They recognize not only the antigens but the antigen bound to major histocompatibility complex or MHC. They are heterodimers of either alpha-beta chains or gamma-delta chains. Cells that express TCRs have approximately 10⁵ TCRs on their surfaces. The T-cell receptor genes are organized into multi-gene systems like that of immunoglobulin. T-cell receptors are also as diverse as the immunoglobulins. Structure. The structure of the alpha-beta or gamma-delta chains of TCRs are very similar to that of immunoglobulin, and therefore they are grouped under the immunoglobulin superfamily of proteins. Each chain in a TCR has two domains containing an intra-chain disulfide bond that spans 60 to 70 amino acids. The amino terminal or N-terminal of the alpha-beta and gamma-delta chains have highly variable sequences, and the remaining of the sequence are conserved. Thus, the TCR domains, one variable and one constant, are homologous to the V and C domains of immunoglobulin. The TCR variable domain has three hypervariable regions that form the complementary determining regions or CDRs. CDRs directly interact with the antigens. TCR also has a CDR4 in the beta chain which does not directly interact with the antigen. After the constant region domain, each TCR chain has a connecting sequence containing a cysteine residue which forms interchain disulfide bonds and held the two chains together in a TCR heterodimer. Following the connecting sequence is the transmembrane sequence of about 21 to 22 amino acids which anchors the TCR to the membrane. This transmembrane sequence in TCR are rich in positively charged amino acids which help them to interact with the signal transducing CD3 complex. Finally, each chain in a TCR also has a cytoplasmic tail of about 5 to 12 amino acids in the carboxyl terminal N or the C terminal. The alpha-beta TCR are predominantly expressed by the T cells in the human and mouse. Organization and rearrangement of TCR genes. The genes that encode the alpha-beta and gamma-delta T-cell receptors are expressed only in cells of the T-cell lineage. The four TCR loci, alpha, beta, gamma, and delta in the germline are organized into multi-gene systems. Each locus has multiple gene segments. The alpha and gamma loci has multiple V and J segments, and beta and delta loci have multiple V, D, and J segments. Functional TCR genes are produced by rearrangement of these gene segments. The alpha and gamma genes are formed by rearrangement of V and J gene segments, and beta and delta genes are formed by rearrangement of B, D, and J gene segments. In humans, the alpha and delta genes are located on chromosome number 14 and the beta and the gamma genes in chromosome 7. The delta gene segment are located between the V-alpha and G-alpha gene segments in chromosome 14. The location of the delta gene segment is significant as a productive rearrangement of the alpha chain gene segment deletes C delta so that in a given T cell, The alpha-beta TCR receptors cannot co-express with the gamma-delta receptors. The human germline DNA contains about 50 V-alpha and 70 J-alpha gene segments and a single C-alpha gene segment. The delta chain gene family contains about 3V, 3D and 3J gene segments and a single C-beta gene segment. The beta chain gene family has about 57V, 2D, 13J and 2C-beta gene segments. The gamma chain gene family consists of 14V, 5J and 2C gamma gene segments. Rearrangement of the TCR alpha and beta chain gene segments result in VJ joining for the alpha chain and VDJ joining for the beta chain. After transcription of the rearranged TCR genes, RNA processing and translation, the alpha and the beta chains are expressed as a disulfide link heterodimer on the membrane of the T cell. TCR germline DNA has conserved heptamer and nanomer recombination signal sequence or RSS containing either 12 base pair 1 turn or 23 base pair 2 turn spatial sequences, flanking each V, D and J gene segments. All of the TCR gene rearrangement follow the 1 turn or 2 turn joining rules, so recombination can occur only between the two different types of RSS. The pre-T cell expresses the recombination activating gene REG1 and REG2. The REG1, REG2 recombinase enzyme recognizes the heptamer and nanomer recognition signals and catalyzes VJ and VDJ joining during TCR gene rearrangement. Allelic exclusion of TCR gene. The delta gene segments are located within the alpha gene loci and during rearrangement of the alpha chain gene, C delta gene segments is deleted. This excluded the rearrangement of delta chain gene and therefore no T cell will co-express alpha beta or gamma delta TCR. Allelic exclusion of TCR alpha and beta chains also occurs. The organization of the beta chain gene segments into two clusters means that if a non-productive rearrangement occurs, the thymocyte can attempt a second rearrangement. This increases the likelihood of a productive rearrangement for the beta chain. Once a productive rearrangement occurs for one beta chain allele, the rearrangement of the other beta allele is inhibited. Exceptions to allelic exclusion are observed in TCR alpha chain genes. Since allelic exclusion is not complete for the TCR alpha chain, there are rare occasions when more than one alpha gene is expressed on the membrane of a given T cell. In such cells, antigen binding specificity is maintained by making only one of the TCR MHC restricted and functional. Generation of TCR diversity. TCR germline DNA contains far fewer V gene segments than IG germline DNA, but several mechanisms that operates during TCR gene rearrangements contributes to a high degree of diversity among T cell receptors. The major mechanisms that generate diversity in TCR are combinatorial joining of variable region gene segments, alternative joining of D gene segments, junctional flexibility, P region nucleotide addition, N region nucleotide addition, and combinatorial association of genes. Combinatorial joining of variable region gene segments. Combinatorial joining of variable region gene segments generates a large number of random gene combinations for all the TCR genes. For example, 50 V-alpha and 70 J-alpha gene segments can generate 3.5 into 10⁻³ possible VJ combinations for the TCR alpha chain in human. Similarly, 57V beta, 2D beta and 13J beta gene segments can give 1.48 into 10 raised to 3 possible combinations of TCR beta chains. Alternative joining of D gene segment. The location of 1 turn 12 base pair and 2 turn 23 base pair recombination signal sequences in TCR beta and delta chain DNA allows for alternative joining of D gene segments. Thus, it is possible for a V beta gene segment to join directly with a J beta or a D beta gene segment, generating a VJ beta or VDJ beta unit. Alternative joining of delta chain gene segment generates similar units. In addition, one D delta can join with another, yielding VDDJ delta and VDDDJ delta. This mechanism generates considerable additional diversity in TCR genes. Junctional flexibility. Gene segments in TCR are joined by recombination. Recombination involves both the joining of recombination signal sequences to form a signal join and the joining of coding sequences to form a coding join. Although the signal sequences are always joined precisely, joining of the coding sequence is often imprecise. This is called junctional flexibility, where the coding sequences can join at different points. This flexibility can generate many non-productive rearrangements, but it also increases diversity by encoding several alternative amino acids at its junction. P-region nucleotide addition. After the initial single-strand DNA cleavage at the junction of a variable region gene segment and attached signal sequence during the joining of gene segments, the nucleotides at the end of the coding sequence turn back to form a hairpin structure. This hairpin is later clipped by an endonuclease. The second cleavage sometimes occurs at a position that leaves a short single strand at the end of the coding sequence. The subsequent addition of complementary nucleotides to this strand, P addition, by repair enzymes generates a palindromic sequence in the coding join, and so these nucleotides are called P nucleotides. Variation in the position at which the hairpin is cut thus lead to the variation in the sequence of the coding join. P nucleotide addition occurs in the gene encoding all the TCR chains and thus generating more diversity. N-region nucleotide addition. Variable region coding joins in rearranged TCR genes have been shown to contain short amino acid sequences that are not encoded by the germline V, D or J gene segments. These amino acids are encoded by nucleotides added during the DJ and B2 DJ joining process by a terminal deoxynucleotide transferase catalyzed reaction. This is called N-region nucleotide addition. The additional diversity generated by N-region nucleotide addition is quite large because N-region appears to consist of wholly random sequences. It occurs in the gene encoding all the TCR genes. As many as 6 nucleotides can be added by this mechanism at its junction, generating up to 5,461 possible combinations. It is estimated that the combined effects of P and N-region nucleotide addition and joining flexibility can generate as many as 10⁻13 possible amino acid sequences in the TCR junctional regions alone. Combinatorial association of genes. Finally, the combination of the two chains in the heterodimer also contributes to TCR diversity. Assuming that the antigen binding specificity of a given T cell receptor depends upon the variable region in both chains, random association of 3.5 into 10 raised to 3 V alpha combination with 1.48 into 10 raised to 3 V beta combinations can generate 5.18 into 10 raised to 6 possible combinations for the alpha beta TCRs. T cell receptor complex or TCR CD3 complex. T cell receptor associates with the membrane protein CD3, forming the TCR CD3 membrane complex. CD3 participates in signal transduction after interaction of a T cell with antigen. It does not influence interaction with antigen. CD3 is a complex of 5 invariant polypeptide chains that associates to form the dimer. A heterodimer of gamma and epsilon chain, a heterodimer of delta and epsilon chain, and a homodimer of 2 zeta chains, or a heterodimer of a zeta and a eta chain. The T cell receptor complex can thus be envisioned as a 4-dimer. The alpha beta or gamma delta TCR heterodimer determines the ligand binding specificity, whereas the CD3 dimer are required for membrane expression of the T cell receptor and for signal transduction. I would like to conclude that the T cell receptors are the major molecule responsible for recognition of antigens by the cells of the T cell lineage. It does not directly bind to the antigen, but binds to antigen presented by self-MHC molecules. It is a heterodimer of either alpha beta chain or gamma delta chain. It is as diverse as immunoglobulin to recognize the varied antigens. The major contributors to TCR diversity are the alternative joining of D segments, junctional flexibility, and the P and N nucleotide addition. T cell receptors are again associated with CD3 complex, which plays a major role in signal transduction.